Okay, so thanks everyone for staying. I have this incredibly daunting and humbling task of trying to summarize this meeting and provide some insights, and I probably spent way too much time on it. So if I exclude anything, and if I say anything that's wrong, let me know and we'll correct it on the slides before they go up. So I'm neither a microbiome expert nor a risk assessor, so this is terrifying for me. So I'll just start by, reminding you what the goals of this meeting were. Obviously, as I said at the beginning, we had a meeting 5 years ago, and this was meant to be an update. Where are we now compared to where we were 5 years ago? So we wanted to have a brief summary and an update, then talk about how does the microbiome actually influence exposure? How does it change our exposure? And then how does it modify susceptibility to environmental toxicants? And then discuss a framework for moving forward. So we began with an update, and we had a very nice talk from Dr. Patterson, who reviewed sort of the concepts in the microbiome. That's not a new idea, as we've heard over and over. And he gave us examples from entomology where we actually understand that the microbiome is an obligate symbiont with many organisms, including ourselves. And they can metabolize all sorts of things and use them as energy sources, including petroleum products and pesticides and so on. So they are incredibly diverse in their functions, and therefore it's pretty much obvious that they're going to change our metabolism. And so the microbiome is outside our body and is all over us, and it actually forms an interface between us and the environment, and as such it modifies our exposures and our dose, particularly the internal dose. So it's going to affect every component of ADME, and so we can't think about dose without thinking about the microbiome. And so one of the questions is, how does the microbiome talk to us? And we heard a very nice presentation on how small molecules functioning probably as weak hormones actually are the communicators, that they go around products of the microbiome and their interaction with us generates small molecules which then interact with receptors like nuclear receptors and FXR. And so that's the mechanism of signaling, and that the modern approaches to metabolomics are the way that we can listen to them talking to one another. And so some of the molecules that were brought out were bile acids and antibiotics and heavy metals are ways in which this happens, and things that we need to monitor. Then we had a very nice talk from Dr. Dominguez Bello, framing the microbiome with respect to us. There's always a question, are we a life support system for them or vice versa? And they were here billions of years before we were. And so basically all eukaryotes have co-evolved with bacteria. And so, we have to live together in order to survive. They occupy every niche of our body. And our immune system is basically developed to deal with the microbiota that we live with. And so that's important because the microbiomes have to are co-developed, and they have to be passed on to the next generation. So that's an important consideration. We heard about how eutherian mammals have developed the need to transfer this on, and this happens primarily during birth, in the birth canal. The uterus is pretty sterile, and so analysis of population structures that have been done indicate that its transmission occurs in the vagina primarily during birth, and this is becoming a problem because C-sections basically disrupt this essential process. So when babies are born by C-section, we don't pass on the microbiome. That's problematic because in many countries 30% of childbirth is now by C-section. That's probably a product of the fact of our evolutionary progress. We have big heads, and because we're bipedal, we have small pelvises. And so, and we're also, that goes along with a reduction in breastfeeding for the most part, and up to 70% of kids are not breastfed, which is another way that the microbiome was transmitted. So the bottom line here is that, our microbiome is being dramatically altered by our medical practices to a large extent. A lot of C-sections are done without the need for, clinical need. And so it's probably pretty likely that by such medical practices that we're actually altering our microbiome and our disease susceptibility in the next generations. So she presents very nice studies where they use a gauze swab that's placed in the vagina before labor and then is used to swab the baby afterwards, and showed very nice data that actually now is not fully, but to a large extent recapitulating or restoring the microbiome to the infant, which is probably a very good thing. And we know that C-section is a risk factor for many diseases. And so this to me was a very exciting finding. Then we talked about evolution of the microbiome in human culture. So the microbiome diversity of primitive cultures is probably very high because they have continuous exposures to different environments. Our built environments are very narrow and very structured, and so the microbiome has been depleted. And urbanization, the effect of urbanization and our hygiene conditions on the microbiome is faster than our ability to evolve. And so we're actually destroying an obligate partner that we need to be healthy. And so that may be one of the factors why chronic diseases are such as asthma and allergies and so on are increasing terribly. So there was a statement made that we really need to go out to these indigenous populations and study the microbiome and understand how different it is from ours so that we don't lose that information before our contact with them basically eliminates that diversity. So then we moved from the populations down to chemistry, and Matt gave us a beautiful talk on an alternative approach to treating the microbiome and dealing with it. And he pointed out that bacteria are the world's best chemists. They make a whole repertoire of chemicals that we can't even imagine or synthesize. And they metabolize almost everything. And he began to look for phase 1 and phase 2 metabolizing enzymes and found that phase 1s are very well represented in the microbiota, but phase 2 enzymes are not there. And so phase 2 enzymes are really a means of secretion in higher organisms for getting the metabolites out of the body. But bacteria need to get rid of those as well. So they have developed, or they recapitulate things like glucuronidases where they can recapture the sugar from these molecules, and that can be a problem for us. When they scavenge the sugar off glucuronidated molecules in the gut, that can reactivate things that we were trying to inactivate. And this was the example he gave was a significant problem that we see with carcinogens and with drugs like anti-cancer drugs and NSAIDs that get glucuronidated. And when they get into the gut, if they're deglucuronidated, then you get high toxicity, and then the doses actually become limiting by the action of the bacteria. And so Matt shows these beautiful studies where he actually developed targeted drugs that affect the bacterial enzymes, but not the host enzymes. So this is a new way of alleviating some of the bad side effects of imbalanced or altered microbiome without actually killing off all the commensals that are good for us. So this, I think, was a very exciting way of looking forward as well. The next session, we got down to the nuts and bolts. How do we do this? And we got an update on all the tools from Dr. Lee, who provided an excellent overview of the current technologies and the state of the art. How do you ask the questions, and how do the questions determine what approaches you use? Whether you use 16S or complete sequencing, how metabolomics and metagenomics get integrated. And he shows some examples of how they're doing this by altering, for example, the composition of the microbiome through infection or treatment with arsenic in one of his studies, or just looking at differences in metabolism across sexes and trying to figure out how the microbiome is contributing to that. And so that was a really nice update on the approaches that we're using and how to best design these studies. Dr. Wilson gave a very interesting talk. I don't know if he's still here or not. But he was talking about how perturbations of the microbiome can be induced by almost anything that we're exposed to. So it's not just the environment, it's drugs, it's foods, it's pretty much everything, our age, and so on. And notobiotic, even being notobiotic affects your metabolic profile. So every time you change anything in your environment, you change your metabolic profile, and therefore possibly your risk of disease. And he also saw very interesting studies that said that if you just look at rats, the microbiomes segregate by the strain, but that turns out to be a big artifact. It has more to do with animal husbandry and so on. So and he gave this very sobering indication that you get strains, cage-specific effects. So you have the same rats and you put them in different cages and you get a different microbiome. So it's housing and individual cages levels even have an effect. And so this is an important thing to take into consideration as we think about moving forward. And he also gave some nice examples of how bacteria, by their normal metabolism, like metabolizing tryptophan, generate a lot of phenols, which then increase our sensitivity to things like Tylenol by depleting sulfation enzymes and substrates. So it was also in the discussion that we focused on the difficulty of relating changes in the microbiome to disease endpoints. There's still a limited amount of data. To do that, we really need to fulfill Koch's postulates, which means take it out, figure out the mechanism, put it back in, and show the same mechanism exists. That's only been done in a few cases at the actual strain level, not for the whole microbiome. And we discussed a lot of issues about experimental design, including how do you monitor and do dosing, duration of the dose, animal husbandry, and the real need for standardization if we're going to do these types of experiments. And we also talked a lot about how we need better in vitro, ex vivo, and notobiotic models and humanized microbiomes in rodents to study and get at the mechanisms. But we also heard that animal models have lots of limitations, and the statement was made that a notobiotic animal is no more like the human population than Justin Bieber is an example of that. And so, I took that a step further and said humanized mice do not represent all, the statement was humanized mice don't all represent all humans. Justin Bieber doesn't even represent all Canadian Justins. So, so the point is that we have to be careful how we use our models so we don't think that this is representative of all Canadians. Then in session 2, we had an absolutely amazing talk from Dr. Elinav, who was Skyped in from Isreal. And his premise was that, obesity is the biggest epidemic that we're facing, and our diets don't work. And even the same diets that are recommended for people give different results. Some people actually gain weight on the so-called diets that are good. And so his concept is that the microbiome is a hub that integrates all the signals from the environment. And so, since we're all individuals genetically, microbiome-wise, and so on, the idea was that maybe we all respond differently, and there is no one diet that's going to help everybody, and that would be true for exposures as well. So, so they initiated the study to look at how diet affects the microbiome and then obesity. Current guidelines for recommendations for food are based on glycemic indexes after feeding in an average of 10 people. He looked at 1,000 people and found that there was more than 1,000-fold variability across people. And so there is no one diet that works for everybody. And so, as I said, he recruited these people and collected an enormous amount of data, all types of metadata on, questionnaires about lifestyle, about, habits, eating habits, and medical records, and so on and so forth. And he called it putting a mirror up to yourself, which I liked. They also did glycemic index every 5 minutes with a pump, and then they did controlled feeding studies and so on, and then used all that data to generate an algorithm which was able to predict for each individual what is your best diet that allows you to maintain a microbiome that actually makes you healthy. And so he found that, for example, that there are vast differences among individuals in response to things like ice cream. So some of us, it's good, some of it's bad. And so, and he also showed, interestingly, that the microbiome was the largest contributor to that variability. So this really shows the power of putting together large cohorts with lots of data, metadata, and using that for modeling, which is something we've discussed over and over. So, of course, as soon as that was over, I called him up and gave him all my information, and this is my good diet that he came up with. So I was pretty pleased with that. There's the ice cream cake. The next session, we focused on the contribution of individual variability, how the microbiome modifies that. And Dr. Shore gave a very interesting talk on asthma. It was news to me that there was a specific type of asthma that was associated with obesity, and that it's non-inflammatory, doesn't respond to typical drugs, and it's actually highly responsive to ozone. So that means that, if this is actually linked to the microbiome, then your sensitivity to ozone can be determined by your microbiome, which was pretty exciting. Interestingly, they took this down to looked at sex differences and so on, but they also took it down to the mechanistic level and showed that it may actually be related to metabolism of bile acids in the gut. so that the products actually go to receptors in the smooth muscle in the lung, and that was the mechanism, which was pretty unexpected and pretty amazing. And then Dr. Garg presented a pretty novel study on using new methods in mass spec to map where metabolites are in the body by taking swabs or taking sections through lungs. And combining that with 16 sRNA, she was able to come up with a sort of cartographic map of where things are. And then look how that's changed by exposures. And so that was a very new, exciting technique to actually look forward and see how environmental exposures to things like cosmetics would change your skin microbiome. And of course, I immediately had that done to myself and was surprised at what I found. Because they were actually showing colors on the skin, so this is what I found myself. So the discussion of this session focused on the role of the microbiome in microbiome data and risk assessment. And the point was made, and we've had this discussion several times now, is that we're already doing a pretty good job of risk assessment with what we know, and that the microbiome is probably being incorporated. But that's kind of an average, and if we want to get down to more individualized risk or interventions, we need to know more about how the microbiome changing over time or in response to exposures, other things like antibiotics. We need to have a better sense of how the microbiome is actually interacting. And so, it's even though we've been doing a good job, we can probably do better. So then Dr. Karagas gave a really nice talk on her epidemiological studies, where she was looking at the effects of early childhood exposures to arsenic, for example, on immuologically related diseases. And so she showed a very nice study from Chile, where a very unique cohort where where moms were exposed, and then that resulted in a higher rate of asthma and wheeze in kids, which was also a new finding. And along with that came the finding that the U.S. has very high levels of arsenic and ends up in rice cereal. Having just gone through having young kids, I know the rice cereal is the first thing you give them, and so we actually give our kids pretty high levels of arsenic if we're not careful. And so the hypothesis was that this might be associated with a lot large incidence of infections that occurs in kids, ear infections and so on, through effects on the immune system. And Dr. Blazer gave an absolutely mind-boggling talk, which was completely comprehensive. The bottom line take-home message for me was that antibiotics early in life have a really terrible effect on your microbiome, and that stays with you. And it affects your rate of obesity. It cooperates with a high-fat diet. And so there's a nice interaction where an environmental exposure, in this case the antibiotic and the diet, actually is synergize and make the disease worse. And if you look at the maps of antibiotic distribution in the US, it maps pretty well with obesity. And then he performed a whole series of very elegant animal studies using genetics and fecal transplants and everything else to show basically that, early life exposures, the effects on the microbiome are reversible, but the effects on the function are not, on the phenotype rather. And so that's pretty scary, in that if we measure something too late, we may actually miss the cause. Okay, and so when we're exposed to things and what effect they have on the microbiome at that time is equally important. And so probably also through the immune system. And the discussion after that session focused on the fact that the microbiome is disappearing, that as it becomes depleted, the next generation receives even a lower amount. And if we keep doing C-sections or exposures and excessive hygiene, that will continue to deplete and further increase our risk of disease that's related to the microbiome. And so there was a lot of discussion about that we should start now and collect samples in ongoing epidemiological studies, and certainly include the microbiome in any studies going forward. There were suggestions made to look at family studies and twins or families with diverse structures, multiple births as compared to single births, and as always, we need larger cohorts and we need more sample collection. But that can become daunting in itself, and there's always a great need for replication in independent studies. And then Dr. Proctor gave a very nice overview of all the ongoings within the NIH and other government agencies on the microbiome. A lot of progress has been made in the last 5 years, and apologize, I just tried to capture some of the things, but there's a lot that needs to be done. And there were some key take-home points here. We need to survey we started surveying the microbiome in healthy populations, and we quickly realized that taxonomic makeup doesn't equal function, and there is no healthy microbiome. And so the phase 2 is much more in-depth, looking at a much more involved and detailed analysis. And then she gave us a list of all the things that have been going on and so on. So I won't go through these for the sake of time. But needless to say, there's a huge effort in the microbiome. It's made it to the president's the union speech, and so it's clearly front and center. So what are some of the next steps that we need to take? So community structure, one of the take-home messages for me is that the community structure does not equal function. Response to stimulus changes over time. So early responses will be transcriptional, later, and the time scales are different. So depending on what questions you need to ask, you need to decide when to take your samples. Horizontal gene transfer is an important part of it, and we haven't even looked at the effect of plasmids and phages and so on. And we haven't really talked very much about farm animals, which is the most frequent source of that. And that was another thing that Dr. Proctor brought out. And there was also a call for development of sequence reference strains for doing functional studies in, for example, notobiotic animals. So some of the kind of take-home messages for me is the microbiome is not static. It incorporates the history of the environment. So that means as our environment is changing and our populations are changing, our risk is probably changing. And that's really an important thing to keep in mind. And as I said, some of these are probably redundant. A change in the microbiome doesn't represent a change in function. Exposures can alter both structure and functions. Changes affect communication between the host and the microbiome, and they affect dose. So the timing and the dosing, the regimen, and the composition of the microbiome all affect the interactions and therefore the dose that's internalized. So a lot more studies need to be done to understand the relationship between exposures and a change in function. What are the tools that we need to use, and which functions are actually important, and what are the mechanisms? That probably has to go down to more of a strain level to individually find out the molecules that are involved. I think for me it was very clear, and I probably said this a couple of times, is that loss of diversity of the microbiome is probably a very risky thing. It's changing our risk to a lot of things. The good news is that risk can be modified through various interventions. I hate the word fecal transplant. I think we should do microbiome transfer or microbial transfer. I mean, I have a hard time talking to anybody and saying we're now doing fecal transplants. It's just an automatic U factor. But it's also dangerous because of the viruses and things that are passages that we don't really know about. So, as we develop more well-formulated transplant concoctions, I think we need to change the name. I think that's important. And then in terms of moving forward, we need to quickly agree on what we should do. What data should we collect? How should the samples be collected? And it's clear, and Yvonne made this point many times, we can't do everything. So we have to be strategic. But I think waiting is a mistake. We have to determine how to best collect these samples. So I think setting up standards and having them available to everybody. So if I have an epi study and I can collect samples, how do I do it? How do I store them? And so on, and preserve them. And we have to be sure we capture the diversity of function, not just of structure. Yvonne made the point that we need to include genetics of the host as well. I think that's well taken. It's probably not a good idea to collect everything because of stability issues and so on. So we need to tailor the samples we collect to the questions that we're asking. And there's an absolute need for standing operating procedures. And again, the population and environment is changing. So samples that were banked a long time ago are useful because as the structure changes and the population changes, it's important to have those historical samples for comparison. We need good in vitro ex vivo models, and we have to be cognizant of Justin Bieber. And we have to determine how the microbiome data can best be used to inform components of risk assessment and decision making. We don't have all the answers, but using modeling, we can probably use some of the information already to try and understand how to move forward, and maybe that will better inform what to collect and when to collect it. And even if we're not sure what data to collect, we need to start doing it soon. And we can, in an iterative way, improve that. 5 years later, if we didn't collect them, we're going to be sorry. And there's modeling, as pointed out many times, modeling has come a long way, and even with limited data sets, we can do some useful modeling. So even if we make good decisions in the past by applying safety factors and so on, we can probably do a much better job in the future. That's kind of redundant. We need to have better coordination in terms of protocols, methods of analysis, and also working with other agencies so we don't duplicate things. And the budgets across agencies should be coordinated so we don't, have hard feelings or things don't get funded properly. And as always, and this has been a theme for the last 6 years in this workshop, is we need better ways of sharing data and cohorts and experiments among agencies to reduce costs and not duplicate. We need to better communicate so that we get the microbiome into new studies like CHEAR. As always, we'd like to encourage collaborations with our industry partners. We'll end on the usual note, which is to encourage consortia and collaborations to better leverage funding for this very important topic. And I'll stop there.